Okay, good afternoon everyone. Welcome to lecture 6 of BMS 300. Today our topic is transcription and translation. And before we get started, I just want to let you know that I updated the slides from lecture 5. There were a couple of typographical mistakes which I fixed. One was in the poll everywhere question. So there was this slide with the answers, with the answer slide on it, and the question was incorrectly written. Two of the options should have been the person is using proteins and fats, but I had written glucose proteins, glucose fats, so I just fixed that. All right, so please download the latest version, and even this morning I uploaded a version of today's slides with a couple of changes as well. So please use that latest version. So here's an overview of what we'll be discussing. Just like how we talked about the various players and the process involved in DNA replication, today we're going to talk about the various players and the process involved in transcription, and we'll see how the mRNA, which is produced as a result of transcription, is going to undergo some modification, and this is known as post-transcriptional modification. And then the mRNA will be converted into protein by the process of translation, and we will see what are the various players involved in translation, but we'll actually get to the process of translation and also post-translational modification when we come back tomorrow. And as we go through these various topics, I'll be pointing out some key structural differences between DNA and RNA, and we'll also look at the various type of RNA as well. We look at 4 types of RNA and how they can play a role in these above mentioned processes. So before we get into today's material, I want to wrap up the poll everywhere question, which I could not get to yesterday. So this is asking, during DNA replication, helicase is needed to unwind the double helix and to pull apart the 2 strands of DNA, right? And so the question is, how does helicase function? Does it function by breaking the hydrogen bonds between the nitrogenous bases? Is it breaking the phosphodiester bonds between the 5 carbon sugars, or is it breaking hydrogen bonds between nucleosides, or is it breaking covalent bonds between nitrogenous bases? So what do you think is the right answer? You can turn to your TAs and neighbors and figure it out. Okay, Kamalani, do you have anyone in your group who has a response for us? Yes? A, B, C, D? Yes, I can't hear you. Okay, hydrogen bonds between nitrogenous bases. Most of you did pick the correct answer, but let me just point out why options B, C, and D are incorrect. So if you break phosphodiester bonds, you'll end up destroying the DNA, right? Because it's this sugar phosphate backbone which forms the which links each of the bases to one another, right? So breaking the phosphodiester bonds won't lead to replication; it'll lead to DNA destruction. And option D is talking about covalent bonds between nitrogenous bases. We know that the nitrogenous bases are held by hydrogen bonds, so that's why option D is incorrect as well. And we see that hydrogen bonds between nucleosides is incorrect because we see that there's a phosphate linked to the ribose sugar and the bases, so they're not called nucleosides, but they are nucleotides. So because of that reason, option C is incorrect as well. Okay, now we move on to the topic of transcription, and you can see that the word scribe is there in in transcription. So what does the word scribe mean? Scribe, to write. So just like how a scribe writes down what is said. Have you heard of medical transcription? The doctor says things, a person doing the medical transcription writes that down. Similarly, the information in the DNA is going to be transcribed into the RNA. So it's the same information. There may be a couple of modifications, but it's just taken from one form and converted to the other. So we find that in transcription, the very first step is the transcription initiation, and this will take place in a region known as the promoter region. So if you look at this figure here, we see that the DNA sequence is depicted like a rectangle, and there is this one point shown as the place where the transcription is going to start. Now if you consider that starting point and you go further upstream, meaning you go further towards the 5' end, that's where you have the promoter region. And in the promoter region, which is upstream to the starting point of transcription, there are a few sequences which are of interest, like the GC box, CAT box, and the importance of those sequences is not within our scope of discussion. I just want to draw your attention to this Tata box shown right here. So the Tata box is a place where you have repeating sequence of thymine and adenines, and the reason I want to point out this sequence is that there will be these transcription factors which bind to the Tata box in the promoter region, and this transcription factors, they are referring to nuclear proteins. So these are proteins which are inside the nucleus. When they bind to this promoter region, specifically in the Tata box, that will cause the recruitment of the RNA polymerase. So notice the RNA polymerase is this really large enzyme which will be recruited, and once the RNA polymerase binds to this promoter region, it will be activated, and that can cause transcription to begin. So the initiation of the transcription is the binding of these transcription factors that will bring in the RNA polymerase, and that can start transcription. So in terms of transcription, we see that there is this step known as elongation, where you have the RNA sequence being produced, and all of this is possible because of one enzyme, and that is RNA polymerase. So when we talked about DNA replication yesterday, we saw that there are multiple enzymes which perform different functions, but the RNA polymerase is like a Swiss Army knife. It has a number of functions it can perform. So one of the first things RNA polymerase does is it's going to cause the DNA to unwind, right? So it causes the DNA to unwind, and then it will start reading the template strand of the DNA from the 3' end to the 5' end. So it will start reading the DNA from the 3' to 5' end, and there is no primer needed in order for the RNA polymerase to function. For DNA replication, you need an RNA primer. Here there's no need for a primer, and we see that the RNA sequence will be synthesized from the 5' to 3' end, just like how the new DNA strand is synthesized from 5' to 3' end. The RNA is also synthesized from the 5' to 3' end, and what the synthesis includes is that the bases which are complementary to the template strand, that is what will be incorporated in the RNA sequence. So for example, if you have cytosine in the template strand, in the RNA you will have guanine bases incorporated, and the only difference is that if you have an adenine in the template strand, in the RNA you'll have uracil rather than thymine. So that's the major difference between, one of the differences between DNA and RNA. And we find that the phosphodiester bonds between these various bases in the RNA will also be catalyzed by the RNA polymerase. Right, so this is how transcription is giving rise to RNA, right? And this is just a very high level view of how RNA can be synthesized. And when we are talking about RNA polymerase, I need to point out there are different types of RNA polymerases. So there are different types of RNA polymerases which will give rise to different type of RNA. Okay, different types of RNA polymerases give rise to a different type of RNA. And what I'm going to do in this slide is just give you a few examples. I'm not going to hold you responsible for what polymerase, RNA polymerase makes which type of RNA. What all you will need to know is what is the functionality of the different types of RNA. But notice that you have this RNA polymerase 1, which gives rise to ribosomal RNA or rRNA. RNA polymerase 2 can give rise to the messenger RNA or small nuclear RNA, and RNA polymerase 3 can give rise to ribosomal RNA, tRNA or transfer RNA, and also snRNA, which stands for small nuclear RNA. And to repeat what I said, all you need to know is that these various types of RNA is produced by different types of RNA polymerases. You don't need to know which polymerase makes which type of RNA. That's a busy detail which you can learn in a more advanced course. And in the upcoming slides, we'll talk about the functions of the 4 types of RNA mentioned here: mRNA, tRNA, rRNA, and the srRNA. So, are there any questions before we move on? If there are no questions, let me ask you a question, right? So this is the format. If you have no questions, I'll ask you a question. So in eukaryotic cells, enzymes that form phosphodiester bonds between nucleotides will function in the blank. Will they function in the nucleus? Will they function in the endoplasmic reticulum? Will they function in the cytosol? Or do you think more than one answer is correct? Where do you think these enzymes which make up phosphodiester bonds between nucleotides will function? You can turn to your neighbors or your helpful TAs and they can assist you. Okay, we have a lot of responses. Gabby, anyone in your group wants to share their thoughts with us? Come on, guys, don't make Gabby pick one of you. Nobody? All right, Gabby, pick someone. We warned them. Yes? Nucleus? All right. Sorry for interrupting you while your mouth was full, but we appreciate it. Yes, so nucleus is the right answer. Ooh, okay. Some of you said more than one answer is correct. So the correct answer is nucleus, and let me explain what. The 2 situations where you can have these phosphodiester bonds forming between the nucleotides is during DNA replication. Yes, are we in agreement? During DNA replication, you have phosphodiester bonds forming between this newly synthesized strand of DNA, between the nucleotides which make up the newly synthesized strands of DNA, and during RNA synthesis as well. As the bases in the RNA are linked one to another, you have these phosphodiester bonds. So we know that DNA is not going to leave the nucleus. It's going to stay inside the nucleus, and so when you have DNA replication, that's going to happen inside the nucleus. And RNA is synthesized from DNA, right? And so those phosphodiester bonds formed between the bases making up the RNA is also going to happen inside the nucleus, right? And more than one answer is not correct. So the only right option is nucleus, and the specific names of these enzymes are the DNA polymerase and the different types of RNA polymerase. Is that clear, folks? Yes? Okay, great. Let's move on to talk about some of the structural features of RNA. So we see that RNA has also got 4 types of nitrogenous bases. You have the adenine, guanine, cytosine, and instead of thymine you have uracil, right? And I told you that thymine differs from uracil in the fact that it has this extra methyl group That's the only difference. We also find that there is this ribose sugar, in that the carbon number 2 will have a hydroxyl group, whereas in DNA it just has a hydrogen group. So it's called deoxyribose sugar, and that's the reason why it's called DNA versus RNA. It's because of that deoxy group. And this hydroxyl group in carbon 2, it makes RNA a lot less stable, because this hydroxyl group can engage in a nucleophilic attack against this sugar phosphate backbone, and that can destroy the structure of the RNA. How many of you here in this class have worked with RNA in a lab setting? Yes, okay, a couple of you. Is it fun to work with? Is it a pain in the neck? Yes. It's very fragile, right? And part of the reason is because of this hydroxylic group, whereas DNA is pretty stable. That's why even from samples in crime scenes, or even from some fossilized samples, you can get DNA as well. All right. So this makes RNA a lot less stable because of that OH group. And we see that all of these bases which make up the RNA, they are linked together by this phosphodiester bonds, right, which make up the sugar phosphate backbone. And because RNA is single-stranded, it's capable of wrapping up on itself and forming some complex secondary structures, right? And we look at some examples of these complex secondary structures in a minute, but I just want to give you a real life analogy. So let's say you're trying to lay down some tape on a flat surface, and you're taking your tape, and as you're bringing it down to lay it down, one hand slips and the tape winds back on itself. How easy do you think is it to unravel that tape? Easy? I think we're better off just tossing that and picking a new piece of tape. So because one side is sticky, it just forms this complex shape, and that's the same reason why the RNA can also form secondary complex structures. But more on this in a minute. I'll just have a quick polar viewer question. The presence of the hydroxylic group in carbon number blank makes RNA less stable than DNA. This should be pretty straightforward and simple. You can just look at the previous slide and answer this, or you can talk to your TAs as well. Carbon 1, 2, 3, or 4. Where is the hydroxylic group which makes the RNA less stable? All right, Justin, anyone in your group has a response for us? Yes? Carbon number 2, correct. Carbon number 2 has the OH group, which can attack the phosphate backbone, and it can make it very unstable. So now let's move on and talk about the secondary structure of RNA. And just like how the analogy I told you, how the tape can stick back on itself and form these complex structures, the single strand of RNA can form hydrogen bonds between the various bases, right? So just like how the DNA strands form hydrogen bonds between the various bases, we see that the bases in RNA can also form these hydrogen bonds between them, and that's why they get these complex shapes. Now, one of the functional importance of these complex shapes is that the RNA can also function as enzymes, and so an enzyme which is made up of the RNA is known as a ribozyme, and we look at two specific examples of how these ribozymes are involved, right, as far as the synthesis of proteins is concerned. So first example is that there is these structures called as spliceosomes, where the ribozymes are present, and they will have a role in editing the messenger RNA. So I told you there will be post-transcriptional modification. These spliceosomes will play a role in that, and they are made up of this ribozymes. Secondly, in protein synthesis, we see that the ribosomes will have a core which is made up of these ribozymes. So this RNA will be complexed with the proteins, and that's what makes up the ribosome, and that plays an important role in the synthesis of proteins. So this is an example of how RNA taking a more complex secondary structure can have a functional relevance in catalyzing reactions. Okay, so now we're going to move on and talk about the 4 types of RNA which we'll consider. Right? We're just talking about 4 because they are relevant to our discussion. The first one is the messenger RNA or the mRNA. This is what serves as a template for protein synthesis. Okay, and we find that the second type is the tRNA or the transfer RNA. And the question is, what is the tRNA transferring, right? So it's going to transfer amino acids to the ribosome, where those amino acids will be linked to one another to form the protein sequence, right? So the tRNA is going to transfer amino acids. How exactly does that? We'll discuss in more detail in the upcoming slides. Thirdly, you have the ribosomal RNA or the rRNA. This is going to form the structural core of the ribosomes, and we'll see the importance of ribosomes when we get to translation. And finally, we have the small nuclear RNA, which is involved in processing both the DNA and RNA, and we'll we'll see an example of how this snRNA is found in spliceosomes when we talk about RNA modification. So on the topic of modification of the RNA, we are going to look at post-transcriptional modification, and we are going to see 3 things which can happen to the RNA molecule once it has been synthesized. So the first thing I want to point out is the 5' cap. This is called 5' capping, and then at the 3' end you have the addition of this poly A tail, also known as the polyadenylation. And finally, you have the step known as splicing, where these intervening sequences known as introns in the RNA, they're going to be cut and removed out, right? So that is known as splicing. What is the relevance of splicing? I'll tell you in a minute, and we'll talk about the importance of each of these modifications in the next few slides. So 5' capping is where you have a modified guanine nucleotide, right? So this guanine nucleotide has a methyl group linked to it, and the way it is connected to this 5' end is through this triphosphate bridge. The name of this group or its structure is something I won't be testing you on. What I will hold you responsible for is knowing what is the importance of having this 5' cap. That's all I need you to know. And there are actually 2 things this 5' cap will accomplish, right? So the 5' cap is going to have 2 functions. Firstly, it's going to protect this messenger RNA from degradation by 5' to 3' exonucleases. So the question is, what do we mean by an exonuclease, right? So exonuclease is referring to an enzyme which will chew up the ends of the nucleic acids, kind of like a Pac-Man, right? So how many of you have played Pac-Man before? Okay, I've never played it, but I think the analogy holds, right? So Pac-Man will be eating up the ends, and similarly these exonucleases will chew up the ends of RNA and DNA. Endo means inside. Endonuclease will cut the nucleic acid sequence in the middle. Exonuclease will chew up from the ends. So this 5' cap will protect the 5' end from 5 to 3' exonuclease degradation. That's the first function. Second function is that the ribosome, it will bind to this 5' cap of the mRNA, and that is one of the first steps in the translation process. So ribosome will bind to the 5' cap of the mRNA. So it is these 2 functions which are accomplished by this post-transcriptional modification at the 5' end of the mRNA. Is that clear? Yes? All right. So now let's look at the 3' polyadenylation, where you have a number of these adenosine monophosphates which are linked. So it's around 50 to 250 adenosine monophosphates are attached, and the purpose is to protect the 3' end from exonuclease digestion from the 3' to the 5' end. So we see that both ends of the mRNA are protected by this post-transcriptional modification. Okay, so we see the importance of two of these post-transcriptional modifications, and now we are going to move on to the third type of post-transcriptional modification, which is mRNA splicing. So I told you that splicing is the process by which this intervening non-coding sequences known as introns are going to be cut out from the mRNA, and these exons, which are the coding regions, they are going to be joined one to another. And all of this is possible because of these structures known as spliceosomes, and I told you that spliceosomes has this snRNA, the small nuclear RNA, which will be complexed with proteins. So because the small nuclear RNA is complex with proteins, it's called SNRPs, or in other words stands for small nuclear ribonucleoproteins, right? So just a mouthful, but that's why we call it SNRPs. So these RNA molecules which have catalytic activity, they're going to be found in the spliceosomes. And so the function of these spliceosomes is twofold. It's going to cut the mRNA, and then it's going to join the cut ends. So it'll cut and paste, no copying. All right, we all like to talk about cut, copy, paste. The spliceosome will cut the mRNA and it will join the cut ends. And so the other term for joining is ligation, and so we see that the intron sequences are removed through the action of spliceosomes. Now there's a question, why are these introns even present, right? If they're not coding for the for proteins, why are they present? Their functionality is beyond the scope of our discussion. Just know that it's going to help with the stability of the RNA, and it can also help in this process known as alternative splicing, where only some exons may be present in a mRNA transcript. So let's talk about the importance of alternative splicing. So notice here that in this DNA sequence you have 5 different exons which are color coded, and then because of alternative splicing you have all 5 present in one transcript, but in the other case you have exon 3 missing, in the other one you have exon 4 missing. Now if you were to not have certain exons, we see that the consequence is that the type of protein which is produced will have a different shape. Now if you have time, you can just count the number of alpha helices versus the red arrows which point to the beta sheets. You notice that the number of alpha helices and the beta sheets, or their positioning, is different. So you see that because of alternative splicing you have different types of proteins which can be made from a single gene. So the analogy I have for you is like if you had an option of buying 4 onesies, or you could buy 4 shirts and 4 trousers, the person who buys 4 onesies has only 4 sets of clothing to wear for different days. But if you had 4 shirts and 4 trousers, you could mix and match, and you can have multiple combinations. So similarly, this alternative splicing leads to many proteins being made from a single gene. Is that clear? Okay. So now let's move on to a are there any questions for me, folks, before we move on? We've talked about transcription and post-transcriptional modification, and I told you why that's important. Yes, what is your question? Could you repeat that? Alternative splicing also? Yes, it could take it out as well. Yeah. We don't get into the functional importance of introns in this course. We're trying to be as superficial as possible to get to the physiology. All right. If you ask me a lot of details on molecular biology, I would refer you to a respective course. Okay, so let's answer a couple of questions. First question is, where are mRNA, tRNA, and rRNA made or synthesized? Is it in the endoplasmic reticulum? Is it in the ribosome, in the nucleus, or in the cytoplasm? Where do you think these 3 types of mRNA would be synthesized? Okay, Izzy, anyone in your group has a response for us? Yes? Nucleus, okay. All right, before we see if this is the right or wrong answer, I have one more question for you. Where do mRNA, tRNA, and rRNA function? Is it endoplasmic reticulum, Golgi apparatus, nucleus, or cytoplasm? And so, again, I'll have someone else answer this. Okay, we have almost 300 responses. Nell, anyone in your group has a input for us? Yes?
Yes, we said
Cytoplasm. Cytoplasm, all right. Okay. So the majority of the class did pick the right answers for both questions, and I want to explain why the answers are what they are by using this figure here. So in terms of the synthesis of mRNA, tRNA, and ribosomal RNA, or any RNA for that matter, it has to happen in the nucleus because RNA is made from DNA, and the DNA is not going to leave the nucleus, right? So RNA will be synthesized in the nucleus. So that's why the question for the answer for the first question was nucleus. Now, in terms of where these different types of RNA function, mRNA, tRNA, and the ribosomal RNA, the answer is the cytosol, because the mRNA is going to leave the nucleus through the nuclear pore, move into the cytosol, and then let's say it's undergoing translation. So the mRNA will be in the cytosol for translation, and it's going to bind to the ribosome, and the ribosome is made up of ribosomal RNA. And where's the ribosome? It's in the cytosol. And the tRNA is going to bring amino acids to the ribosome, and this happens in the cytosol where the amino acids are linked one to another to make the proteins. Right? So that's why these three types of RNA will function in the cytosol. Now, if I had included small nuclear RNA, then what do you think the answer would be? Nucleus, right? Small nuclear RNA is going to function in the nucleus because the splicing of the mRNA is happening inside the nucleus. Right? So I did not mention small nuclear RNA because I didn't want to confuse the issue. All right, so now let's move on and talk about translation. Right? So translation is the process by which the messenger RNA is going to be used to make the specific type of protein. And the very first step is that the messenger RNA has to be read, and I need to tell you how the messenger RNA is going to be read. And it's going to be read from the 5' end to the 3' end, and it's going to be read 3 bases at a time, right? And so we see that these bases are read in triplets, and the name for these triplets is known as codons. And I told you we have around we have 20 different amino acids, and if you were to take the 4 bases and to have just 3 coding for a type of amino acids, if you look at the number of combinations, we get a total of 64 combinations, right? And so what you see in this figure here are those 64 combinations of those codons. And we see that because there are only 20 amino acids, it is not surprising that each codon will code for a single amino acid, and most of the amino acids have multiple codons. That's how you can have these 64 codons coding for 20 amino acids. So I want to draw your attention to this example here. Notice here that threonine is coded for by all of these 4 codons, but if you look carefully, the first 2 bases are identical, right? It's only the 3rd base which is changing, right? And so the fact that this 3rd base alone is different will help us to understand this concept of wobble hypothesis. So the term wobble in common English is referring to something not being steady. You could say, look at the toddler, she's wobbling as she walks. But what does it mean as far as wobble hypothesis is concerned, right? So we see that the wobble hypothesis is referring to, it's explaining how the tRNA is going to interact with the mRNA. Okay, it's explaining how the tRNA or the transfer RNA will interact with the mRNA. We see that the tRNA has got two things I want to point out. It's got a number of features, but there are two things which are important to our discussion. The first is that there is an amino acid carried by this tRNA, right? And this amino acid which is carried by the tRNA is going to correspond to the codon to which that tRNA will bind. Okay, first thing is the amino acid which is carried by the tRNA will correspond to the codon to which that tRNA is going to bind. So the next question is, how does this tRNA find where the codon is, where it should bind? And it's because it's got this anti-codon sequence which is going to be complementary to this codon sequence. And so the second thing is that the anti-codon sequence will help the tRNA to find where its corresponding codon is. And so in the interaction of the anti-codon with the codon sequence, only the first two bases need to match precisely. The third base may or may not match, right? That's what this wobble pairing is saying. Even though you don't have this third base matching exactly, if the first two match, the specific tRNA can find that codon which it is looking for. So we see that there are 64 different codons, right? But there are only 40 tRNAs, because a single tRNA can be identifying multiple codons, because only the first two need to match and the third thing may or may not match. And so this wobble pairing is where the third position is not having an exact match. Examples of this could be that guanine could base pair with uracil. Remember that guanine is a purine and uracil is a pyrimidine, and so even though there isn't an exact match, they can still base pair. So this is the first thing I need you to know about how the mRNA is read in triplets and from the 5' to 3' end. Okay, now let's talk about the open reading frame, since we're talking about how the mRNA is read. So the open reading frame is referring to the coding region of the mRNA, and it's important that the starting point is identified correctly, right? So the starting point of this reading frame has to be identified, and this happens because of a start codon which codes for AUG. So AUG is also coding for methionine, but when it is first encountered in the mRNA, it's going to indicate the starting point of the reading frame. And there are these three codons which are known as stop codons, and they will indicate when the translation should terminate. So if any of these three sequences are encountered, UAA, UGA and UAG, that indicates that the translation is going to terminate, because once these sequences are encountered, there will be these release factors which bind to the ribosome, and that will terminate the translation. Exactly how this happens we will discuss when we talk about the steps involved in translation. And I'm not going to hold you responsible for memorizing these codons, but if any of you are going to work in a lab and you ever are in a need to remember these, here's a fun way to remember it. UAA stands for you are annoying, UGA is you go away, and UAG is you are gone, right? So anyway, you may never use this, but at least helps to wake up people if they are feeling drowsy in the last 10 minutes of class. Alright, so while we are on the topic of the reading frame, I need to tell you how mutations can have an impact on the sequences which are being read, right? So an example of this is a point mutation, where a single base is going to be added, or it could be deleted, or third, it can also be changed. So any of these changes to the base could indicate that a different type of amino acid would be present in that place where a mutation occurs. Now in the case where you have an addition or the deletion of a specific base, it can lead to what is known as a frame shift mutation, where the reading frame from that point onwards is going to be completely different. So example, notice here that you have CAA, and instead of that A, a cytosine is inserted in, right? And so this A is now pushed, and so from that point onwards, notice that all of the amino acids which are going to be encoded from that point onwards is going to change. So it's caused by a shift in the reading frame, and that's what is known as a frame shift mutation. Now I want to give you a few other examples of mutations as well. And here is an example of a missense mutation where it was formerly UAC, which coded for tyrosine, but then now because it is UCC, it is coding for serine, right? So these names may not mean much to you, but I want to give you an example of how a change in a single amino acid can have fatal consequences. And an example is a mutation which causes hypertrophic cardiomyopathy. The difference between these two amino acids was just a single methyl group, and that led to hypertrophic cardiomyopathy, where the heart muscle was pathologically remodeled so that it became very thick, right? So even a small change in the protein structure can lead to functional differences. And then you have these nonsense mutations where the change in the base could incorporate a stop codon. So notice that this makes the protein to be a lot shorter. It's going to be truncated prematurely because of a stop codon which is introduced in. And hopefully the best case scenario is where there's a silent mutation, where even though there's a change in the base, the type of amino acid which is encoded by this modification is the same, right? So assume that this modification happened at base number 3, and the type of amino acid encoded was no different, right? So that's the best case scenario. Now I have a question for you to make sure you all understand the types of base pairing, and mutations. So here's the question: a mismatch between the third base of the codon and the anticodon is referred to as blank. Is it a point mutation, a missense mutation, wobble pairing, or frame shift mutation? What do you think it is? If you have a mismatch between the third base of the codon and the anticodon. Okay, we have lots of responses. Okay, Justin, anyone in your group has a response for us? Wobble pairing. Okay, should be pretty straightforward. Most of you did pick wobble pairing, and that's what this is referring to, where you have a mismatch between the third base of the codon and the anticodon on the tRNA. So now that we have talked about the mRNA, that's the first player in translation, right? We're going to talk about the other key players in translation. The various steps involved in translation, we'll get to it tomorrow, but let's just wrap up today by talking about the other players involved in translation. So we see ribosomes play a very important role. Notice that the ribosome is going to read the mRNA sequence, and then it will cause the respective amino acids to be linked with one another. And we find that this ribosome has got the RNA, which is complexed with proteins to form the core structure. And you're going to see what are some of the key features of the two subunits which make up the ribosome. So you have a smaller subunit and you have a larger subunit, and let's look at their key features. Now the small subunit, it's the one which first binds the mRNA. And which part of the mRNA does it bind to? It binds the 5' cap, and it will start reading the mRNA sequence. And the moment this small subunit encounters the start codon, it's going to recruit the larger subunit to come and bind, and thereby it will make this the ribosome complex. Now let's see what are some of the key features in the larger subunit. So in the large subunit, we notice that you have these ribosomal RNA which are linked with associated with proteins, but there are two things I want to point out. Firstly, you have three sites. You have the A site, P site, and E site for the docking of the different tRNA. So tRNA is bringing the amino acids to the ribosome, so there are docking sites for the tRNA to bind. And secondly, this larger subunit will help to form those peptide bonds between the amino acids. So all the amino acids which come in, the peptide bonds being formed between those amino acids is because of this larger subunit. Now let's talk about these three docking sites. It's called the A site, P site, and E site. It's very nice that it all spells ape, right? So it's easy to remember these three sites. Now each of these have a functional relevance. So the A site is where the tRNA is going to arrive with the amino acid linked to it. So this is the site of arrival. P site is where the polypeptide chain is going to grow. So the protein sequence is going to grow as the amino acids are linked to it. And the E site is where the tRNA, which has given up its amino acid, is going to exit, right? So A for arrival, P for the growth of the protein chain, and E for the exit of the tRNA which has given up its amino acid. So we've talked about the mRNA, and now we talked about the ribosome components, and now we're going to move on to the third player, which is the tRNA. And we already briefly talked about the tRNA in the previous slides, but let me quickly review a couple of facts. So we see that the tRNA is carrying the amino acid, but it also has a specific anti-codon sequence, and it is this anti-codon sequence which will bind to the codon sequence because of the complementary basis. So here's an example. If the codon was AUG, then the anticodon, when it's read from the 3' to 5' end, would be UAC, and if you were to look at the anticodon from the 5' to 3' end, it would be CAU. And so it's because of this complementarity that the anticodon can bind to the codon sequence in the mRNA. And once the tRNA is binding to this mRNA, notice that it's going to give up its amino acid to this growing polypeptide chain. How exactly this happens, we will discuss when we come back tomorrow. But the main question I want you to ask yourself is, how does this tRNA get its amino acid? So once it gives up its amino acid and it leaves, how is it going to be linked to another amino acid? And so this is possible because of this large enzyme known as the aminoacyl tRNA synthetase enzyme. And because we have different types of tRNA and different amino acids, you have different types of these tRNA synthetase enzymes. And notice that there is a binding pocket for this empty tRNA. There's a pocket for ATP to bind, and also for a site for that respective amino acid to bind as well. And so this is an energy intensive process where the bonds between the energy, those phosphate bonds in ATP, will help to link the amino acid to the tRNA, and this process of the amino acid linking to the tRNA is known as the charging of the tRNA, and then the tRNA can go drop off its amino acid. So let me summarize what we discussed. We talked about how the RNA is single stranded, but it can form pretty complex shapes. We talked